will receive, and that's how the actin filament moves from one side to another. It polymerizes and depolymerizes at the different endings. For that, there are actin-binding proteins that have different activities. One that allows actin to polymerize in a faster way, like, for example, profilin. Another one, thymosin, particularly beta 4, but thymosin in general, slows down the polymerization or inhibits the polymerization. That means that that end will recede instead of being growing. This is an example. We have profilin here, rapid plus N growth, because it helps to speed up. But here, with thymosin, it helps to sort of slow down. Okay? Also, there are actin binding proteins that are involved in severing the actin filament, not only from the ends, but also from the middle, like gelsolin. This is a typical example of an intermediate filament, lamin here, which is responsible for binding and for tightening up the nuclear envelope, the inner membrane of the nuclear envelope with the nuclear pores here with the DNA. Plays multiple roles. It has multiple binding sites. It binds to many aspects of cellular contents here and tightens up all these components. During mitosis, lamin gets phosphorylated and degraded. And since it loosens up, the membrane sort of also disaggregates, and that allows for the mitotic spindles and mitotic procedures. You don't need to learn all these names, but just to have the concept of what's going on. Is another example of intermediate filaments. These are two cells, one and the other one, that are connected at the desmosome, which are plaques here with giant proteins here that forms filaments. For example, vimentin is one of those. There are many others. And we'll see all that in subsequent lessons. All these structures help the cells to be strong, very strong, tightens up the binding of the cells. Okay? Within the different cells and inside the different cells. These are the intermediate filaments. And this is the tubulin, or the microtubules, made out of two isoforms of tubulin, the alpha and beta tubulin. They form a heterodimer, and they align in a very oriented way, forming these spirals here, and leaving a hollow area inside and lumen. And you can see an electron of the microtubule, again made out of two isoforms, beta tubulin and the alpha tubulin. Okay? And this is another view. This is a cross-sectional view with the microtubules here, with the hollow area. This is the microtubule, the microtubule, and these are motor proteins that utilize energy to walk along the microtubules. And they are very large complexes, like the kinesin with the kinesin receptor, many other complexes that bind to a vesicle with a cargo inside. So the vesicle with the cargo walks along the microtubule. It's not like it swims within the cytosol. It walks along the microtubules. And it goes from the minus n to the plus n, for example, in this case. Here you can appreciate how complex this can be, and this is a simplified version of the dynein complex that go towards the minus n of the microtubule. So this is dynein, and it associates or binds strongly to another set of proteins and subunits. Spectrin, all these are huge proteins, very, very large complexes, and the proteins are very large. Ankyrin and many other proteins that anchor the vesicle and the cargo. Okay? Glycoprotein inside of the lumen. Right? Never a glycoprotein outside of the lumen. Okay? And this dynein walks along the microtubule. This is an example to show you how regulated is the process of cell sorting. It's not just random that the vesicle pinches off and then goes wherever it has to go. It's all tightly regulated. And mitosis, very, very complex and regulated process, which starts with the disaggregation of a nuclear envelope by phosphorylation of laminin. Okay? But the nuclear envelope disappears, and the microtubules organize, forming spindles, chromosomal spindles. The DNA organizes and condenses at the metaphase period. Then the nitrate is segregated, and then the cleavage furrow that separates the two cells, and then the two nuclear envelopes that conform the two nuclei. Right, we're going to stop here tonight. Remember that this is another view of what's coming.